Greetings to all my viewers. This is Dr. Raktim Mukherjee, Associate Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University, Anand. This is the second video in the series of cells and organs of the immune system. In the first video, we already saw the process of hematopoiesis and introduced ourselves to the various blood cells that are formed as a result of hematopoiesis. To be precise, the lymphocytes, the granulocytes and the agranulocytes. In this video, our major focus shall be on the innate immune cells. However, before we go to the exact details of innate immune cells, let's break the immunology ice by connecting my previous video with the current one. To get into the overview, the immune system consists of diverse cells. When I say diverse cells, there are several cells. This constitute the cells of the immune system, and these cells, they work together to protect the body against pathogen. Further, these cells originate from the HSCs, as we saw in the previous video, the hematopoietic stem cells in the bone marrow, and they thereafter mature in various tissues. Let's recap the process of hematopoiesis that we studied in our earlier video. All immune cells originate from HSCs or what is called as the hematopoietic stem cells. A part of the hematopoietic stem cells will lead to its auto renewal so that the pool is maintained. This hematopoietic stem cell is further differentiates into two major progenitors, the common myeloid progenitor, which we talked earlier as CMP, and a common lymphoid progenitor, which we shall abbreviate it as CLP. These progenitors are the source of various immune cells. When you talk about the common myeloid progenitor, it gives rise to erythrocytes, your RBCs, the mast cells, the mediator of inflammation, and the myeloblasts. Further, it gives rise to megakaryocytes, which ultimately leads to thrombocyte formation. Megakaryocytes we already dealt in our previous video. Myeloblast in turn gives rise to the three major granulocytes. They are called as granulocytes because they contain granules. And these granulocytes are basophil, neutrophil and eosinophil and monocytes. Monocyte, once it migrates to the tissue, it will be called as macrophages. I will abbreviate it as M5, that is macrophage. Going to the common lymphoid progenitor. Common lymphoid progenitor differentiates into NK cells, the natural killer cells, and the small lymphocytes. Talking about small lymphocytes, these are the T lymphocytes and the B lymphocytes. As we know that the B lymphocytes are the arms of humoral immune response. They differentiate into plasma cell, which produce the antibodies required for pathogen neutralization. Now we come to innate immune cells. The following are the major innate immune cells: the neutrophils, the macrophages, the dendritic cells, and natural killer cells. In this video, we shall study all these cells in greater details. But before that, let's have an overview of all the innate immune cells. As I said, first is neutrophil, followed by macrophages. First is neutrophil, second is macrophage, the dendritic cells, and the natural killer cells. We will take up just a brief overview of each of these cells, followed by the details. Neutrophils, they are the most abundant phagocytes. These neutrophils engulf and destroy pathogen via the process of phagocytosis. In very layman's language, I call it as hug of death, because the immune cell, the phagocyte, will extend its pseudopodia or the arm and engulf the pathogen with hydrolytic enzymes. Macrophages, the phagocytic cells, are present in tissues. The dendritic cells, these macrophages act as scavengers or as APCs or antigen presenting cell and effector cells. Dendritic cells, they are the bridge between the innate and the adaptive immunity. These dendritic cells capture antigen and present them to T cells. Natural killer cell, they recognize infected and cancerous cells and release cytotoxic granules to induce cell death. These were some of the brief overview of each of the innate immune cells that we shall acquaint in this current video. Now in greater detail. First we go with neutrophil. This is the diagrammatic overview of neutrophil. You can see that the neutrophil is has a multi-lobed nucleus. You can see multiple lobes. If I count 1, 2 and 3, the multi-lobed nucleus. Okay. These are the 3 lobes. Further, if you see, the neutrophils has granules, and this is the reason the basophil, the neutrophil, because of the presence of granules, they are called as granulocytes. And in fact, you can find phagosome because neutrophils also act as phagocytes. Later, after the pathogen is engulfed, the phagosome fuses with the lysosome to form phagolysosome. So neutrophils are the abundant white blood cells. They are the first line against infection, and this is the reason we ascribe them to innate immune cells. They are phagocytic. They engulf and digest pathogens. The neutrophils has granules with antimicrobial peptides leading to the clearance of the pathogen. Further, the neutrophils play a vital role in the early stage of inflammation. When I say early stages of inflammation, they in turn recruit other immune cells. These other immune cells, they will come to the site of infection. They will go to the site of infection and leads to the pathogen clearance. Talking about the lifespan, these neutrophils are very short-lived. That is, the lifespan is very less. In fact, they would die after few days of activity. I'll write down that they die within few days, and during these few days of activity, they form a significant part of pus at the infection site. So whenever someone asks you which immune cell is involved in pus formation, your answer should be neutrophils, which are a component of the broad, that is leukocytes or WBCs. Now when you talk about certain inflammatory conditions, the neutrophils can cause tissue damage due to the release of reactive oxygen species. Reactive oxygen species are also abbreviated as ROS. Mind you students, these reactive oxygen species also have a very important or prominent role in immunology. Although they cause collateral tissue damage, they in turn help in clearance of pathogen. The neutrophils further contribute to the resolution of inflammation by promoting the removal of cellular debris. So once they remove the cellular debris, that is all the junk material, they will help in resolution or inflammation. Our next cell type is macrophages. The macrophages are versatile phagocytes, that is, they are found in various tissues throughout the body. So when you talk about versatility, it means a person like having multiple talent. So here also it is versatile phagocyte, that is, they are found in various tissues. They serve as the first responders to scavengers, that is, the dead cells and the debris. So whenever you have some of the cell which are dead or you have debris, the macrophages are the first ones to respond. The macrophages are also involved in antigen presentation, that is, they display the digested pathogen fragments on their surface and they in turn activate the T cells. As you can see, they in turn leads to activation of T cells. If you see the diagram, this is your macrophage. You can find so many pseudopodias which helps the macrophage to act as a phagocyte. There are phagosomes, there is a lysosome. Later on, the phagosome and lysosome would fuse to form the phagolysosome. This is what it is. After fusion, you can find the phagolysosome. This is where the hydrolytic enzymes will act on the pathogen. Further, the macrophages recognize diverse pathogen recognition receptors. They release cytokines for regulation, and amongst the cytokines you have M1. M1 group of cytokines are pro-inflammatory, that is, they promote inflammation, and M2 group of cytokines are anti-inflammatory, that is, they will negate the effects of inflammation. So these states are context dependent. They also function as tissue specific macrophages in various tissues such as the Kupffer cells in liver and microglial cells in the brain. In my next slide, I will brief you about the various tissue specific macrophages. So the various macrophages along with their functions and locations, something which we already saw in our earlier video. In liver, it is Kupffer cells, lung alveolar cells, spleen, the marginal zone macrophages and red pulp macrophages. Bone, it is osteoclast. Next we go to the dendritic cells. You can see the diagram of dendritic cells. Here also you have phagosomes. There you have the phagosomes, the various process, and here also you have a very prominent lysosome. Some of the important points of dendritic cells. The very first, the dendritic cells are crucial in linking the innate to the adaptive immunity. So there's a link between the innate and adaptive immunity, that is dendritic cells. What the dendritic cells do, they capture the antigen from pathogen. Thereafter they process them, and after processing they present the antigen to the T cells. Once they present the antigen to the T cells, they will initiate the adaptive immune response. So what I would do, I will highlight T cell presentation. After T cell presentation, this will be the prelude for adaptive immune response. The immature dendritic cells are very specialized. They are very specialized in capturing antigen at the site of infection. This is about the immature dendritic cells. Whereas the mature dendritic cells migrate to the lymph node to activate the T cells. So mature dendritic cells will migrate, they will migrate to the lymph node, and once they migrate to the lymph node, they in turn will activate, I will write activate the T cells. So you have immature dendritic cells which captures the antigen, and you have the mature dendritic cells which will migrate to the lymph node to activate the T cells. The dendritic cells express various PRRs, that is pattern recognition receptors. These pattern recognition receptors help the dendritic cells to detect pathogen, and after the detection of pathogen there will be initiation of innate immune response. Mind you students, our current topic is innate immune cells. The dendritic cells also possess a long branched extension. See, if you see the diagram, you can see processes. These are the long branched extension, and these extensions are called as dendrites. The branched extensions are called as dendrites, and these dendrites increase the surface area for the antigen capture and interaction. So once the surface area increases, the more antigen will be captured and further interaction would take place. The dendritic cells are central to immune surveillance, that is, they act like a security guard, continuously doing surveillance, and they play a very pivotal role or very important role in maintaining immune tolerance by inducing the regulatory T cells. So they are central to immune surveillance, and they will induce immune tolerance. How they will induce immune tolerance? By inducing the regulatory T cells. I will write TREG, that abbreviates T regulatory cells. So they are the central to immune surveillance. Whenever there is a dysfunction of dendritic cell, this dysfunction can lead to autoimmune disease. It's a separate topic altogether from immune system, and this autoimmune disease will have impaired immune response against pathogen. Now we come to the last innate immune cells, that's the natural killer cells. If you see the diagram, the natural killer cell has activating receptors and inhibitory receptors. They help the natural killer cells in controlling. The natural killer cells are the subset of lymphocytes which are exclusively known for their ability to recognize and eliminate infected or cancer cells. This it does even without prior sensitization. So that is very important to protect the host against cancer or tumor formation. They will provide very rapid response against viral infections. So rapid response, they will provide rapid response against viral infections, and they contribute to tumor surveillance. The natural killer cells have inhibitory receptors, as shown in the figure. There you have the inhibitory receptors. So these inhibitory receptors will recognize self molecule, and thus the natural killer cells won't attack your host cell, thereby it will prevent autoimmune disease. However, the same natural killer cells, when they encounter the cells that lack the self molecules or display stress markers, the natural killer cell will be activated, and thus it will protect the host, but it will give a death blow to the pathogen or altered self cell. The natural killer cell has several cytokine like interferon gamma, which will enhance the immune responses. They have perforins, the granzymes. They in turn will lead to apoptosis in target cells. So someone asks you how natural killer cell induce apoptosis. It will be through the perforin and granzymes that they carry, which will drill a hole and lead to the perforin granzyme mediated apoptosis. They will produce interferon gamma, which will enhance immune response. Further, there is a subtle balance between activating an inhibitory signal, which helps in a fine tuning or control of the natural killer cell, that it kills the pathogen or altered cell without causing any damage to the host cell. In certain conditions such as viral infections and cancer, the natural killer cells can become more active to control the spread of infected or malignant cells. So students, it's not that we don't develop small tumors. Time and again it happens. Even we don't notice that it's happening because our natural killer cells are continuously doing surveillance and it kills the tumor cells and prevents from cancer formation. So those were some of the details of the innate immune cells. In our next video, we will see some of the adaptive immune cells. To conclude, the innate immune cells are initial defense pathogen recognition. They are the initial defense against pathogen recognition. The innate immune cells are the body's first line of defense against infection, with crucial role in recognizing and eliminating pathogen. The neutrophils, the macrophages, the dendritic cells and natural killer cells, they all contribute collectively to rapid immune response. These innate immune cells play diverse role from phagocytosis to functioning as APCs or antigen presenting cells. This way, they directly target and kill the pathogens and regulate the immune system. They function in a coordinated fashion. Their coordinated actions ensure immediate protection while also influencing the development of adaptive immune responses. In the next video, we shall highlight the adaptive immune cells. Till then, thank you very much from Dr. Raktin Mukherjee's World of Biology.